Hi, my name is Richard Rachman. Today I'm going to be talking about tree rings and disappearing water, research on sagebrush in Owens Valley. I'm a plant ecologist, graduate student, and I'm the biodiversity coordinator for the CSUN Institute for Sustainability. So the first part of my talk of what I want to address is a land acknowledgement. Uh, this is the land of the Numu of Payahunadu, also known as the Owens Valley Paiute. Um, this land historically and continues to be pillaged by white settlers, and it's important to address that. Uh, Owens Valley was named after this man named Owens, who was never in Owens Valley, but he had a friend name the place after him. And so there's been a lot of really egregious stuff that's happened in this valley, and it's important to address that again. So a very small thing that you can do, especially if you're from Los Angeles like myself, if you do visit this place, make sure you support Paiute businesses and be respectful of the land because we really are guests on it. And literally Los Angeles would not be what it is without Owens Valley. And it's important to acknowledge that and try to do the best you can to correct that. So today I'm going to be talking about my research about Owens Valley, and I'm going to be talking in the style of kind of a scientific presentation, right? So it's going to be an introduction into this land and the research behind it. Then I'll go into some of the methodology behind how we did our research. And then finally, the results that we found from this, discussing those results in our discussion, and then some concluding remarks that we can say. Let's jump right in. So we're going to be talking about this place called the Great Basin. Now, the Great Basin is one of the great deserts of the United States and North America. It's gigantic. It covers so many different states. And it's really dominated by sagebrush, one of the species of which is Artemisia tridentata. And it has a really big range across North America, going all the way down into Baja California. But part of the extent of that range goes into Owens Valley. And that's mainly what we're going to be focusing on today. But part of what makes the Great Basin so unique as a desert is it has these cold, snowy winters and then these hot, dry summers. So there's a really big shift in temperature that goes on between the winter and the summer. And that's an important part for the ecology of this shrub. So I mentioned we're going to be talking about Owens Valley, right? So where is Owens Valley in California? It's kind of eastern California in the central part. And then 200 miles down south is going to be Los Angeles. And we have a really integral part relationship with Owens Valley, where LADWP has bought out a ton of land in Owens Valley. And that's where a lot of our water comes from, where it goes fed by gravity from Owens Valley all the way down into the San Fernando Valley into Los Angeles. So how did we come upon this research, right? So this started in 2014, 2015. My advisor, Dr. Polly Schiffman, was doing research in Owens Valley, looking at how the vegetation communities have changed over time and how that affected people that lived in the area, right? So it's kind of this collaborative effort between anthropology and botany. And so one thing that they noticed was there's a lot of dead sagebrush. This is a really long-lived organism. Some individuals in certain parts of the state can get over 200 years old. Most sagebrush is like around 100, maybe younger, but that's still a long time. And so seeing these massive populations in Owens Valley dead for seemingly no apparent reason, it should cause concern. And as this is the southern extent of the population, it's going to be a little more sensitive to climate change, right? I mentioned that sagebrush is adapted for these cold, snowy winters, these hot, dry summers. So if something messes with that, then this population could be more easily affected. So let's talk about some of the politics and history behind Owens Valley. So this picture here is where the LA aqueduct actually starts and goes in Owens Valley. And so this is the water that eventually makes its way into the um, lawns and homes of San Fernando Valley and a lot of Los Angeles. So in the 1800s, what happened was white settlers colonized Owens Valley and stole land from the Paiute, and pushing the Paiute out of their ancestral homelands where they had complex agriculture and really destructive force in the region. And then in the early 1900s, LADWP saw an opportunity where they needed water, where they perceived that they needed water because of a population explosion. And so what they thought and what they did was they bought the water rights and the land rights from under these farmers, and they took them for themselves. And throughout the decades in the early 1900s, they bought more and more land and water legally with a lot of endorsement from the US federal government, which also owned a lot of land in that region. And so they then built a large aqueduct from the Owens Valley 200 miles south, fed by gravity, and a really engineering marvel. Brought all this water down from the Owens River, from Owens Lake, and even from the water table through a series of wells throughout the Owens Valley, brought that down to Los Angeles. And so it's not just the Owens River, it's also this lake, this massive lake that used to exist within Owens Valley has been almost completely dried up because of this water being taken from this region to create a metropolis that wouldn't have existed otherwise. So what are the effects of this, right? When you're taking so much water, here's a picture from Owens Lake. And as you can see, it's weird to even call it a lake. And historically, this road would not even been able to be there because there would have been so much water. So what happens when you take all the water from a lake? Well, those sediments that would be under pressure from the water are then going to be exposed to air. And that dust that normally wouldn't see the breath of air would now be exposed and be able to be kicked up as dust. And it's created some of the most toxic air quality in the United States. And this dust kills a lot of people, causes cancer and all sorts of terrible disorders. And there's been lawsuits against LADWP, so they have to keep a certain amount of water on the lake bed surface. The area is very hard to visit. It's definitely a scar on the landscape of a lot of pain and suffering by people. I suggest everyone from Los Angeles go visit at least once. So, but this has also caused a lot of economic struggle, not only from like medical bills and such, but when you remove water from a region, it really limits the amount of farming or type of farming that folks can do there. Whereas with the Paiute have like for a really long time have had really complex agriculture in that region themselves. A lot of the people that then in the 1800s and 1900s that were farming there, they would be doing fruit trees and alfalfa. This has impacted them and how much water that they're able to irrigate their crops and farms with. So that's had like an economic problem where a lot of people have just like left because of it. And of course, like a lot of the Paiute like that are on their reservations, like they don't have as much of that social mobility to be able to just up and leave. It's their land, right? So this has impacted the reservations as well and what they can do in terms of economic stability. This water sequestration not only affects crops, but it could be impacting vegetation as well, right? And to someone that lives there, this is pretty obvious that if you're taking a ton of water from the land, what might be once like a really lush green landscape is then slowly going to, through a process of desertification, change and turn to a lot of scrub, right? So how is this water sequestration possibly impacting the vegetation is an important question. One example of how changes in vegetation type affect animals is sage grouse, and specifically the bi-state sage grouse, which is a very rare metapopulation of sage grouse. It's kind of extravagant looking birds, right? Where the males have these like really beautiful plumage and these large gular sacs that they use for long distance communication, and they compete for female attention. they're really at risk because of like natural gas extraction, because of habitat fragmentation of roads and power lines and ranching. These sorts of things affect their habitat. And sage-grouse need sagebrush. They are sagebrush obligates. They need to eat sagebrush at certain times of year. They need it for nesting. They need it for habitat, for hiding. Sagebrush is an integral part of the sage-grouse's natural history. And if you're destroying populations of sagebrush, this is going to severely impact this bird, which was once so plentiful that people could depend on it for a food source, becoming more and more rare and in need of conservation. So sagebrush is important as a habitat type for a lot of animals. And so we can look at sagebrush and how it's being impacted by stress, right? But so what sort of stress does sagebrush deal with? So sagebrush, despite living in a cold desert, right, it has a pretty low water use efficiency. What does that mean? And this is the ability for water to not evaporate away from the leaves. A lot of when sagebrush gets hot, it kind of has this ability to just evaporate tons of water. And this is different than a lot of other desert plants, which might be a little more conservative with their water use efficiency. So this makes sagebrush uniquely susceptible to drought stress, right? Another part of sagebrush, though, is the way that they deal with this is they heavily depend on groundwater in order to become hydrated, right? So water that's underneath that you may not be able to see, that's in the water table. So sagebrush have pretty deep roots, about 2 meters deep. And if that water is not there, this can make them even more drought susceptible, right? And to drought stress. So it's really important that they be able to reach the water table and to be able to extract that water from it. So then how do we see the stress in sagebrush that this drought, these dry conditions can possibly bring? So through the wood. And so there's this form of science, tool in science called dendrochronology. That's dendro for tree and then chronology for time. So we can see the growth in wood over time. And dendrochronology is an important field for studying climate. You can verify the age of a violin, Stradivarius violin, through looking at the tree rings. We've been able to do paleo reconstructions using dendrochronology for what conditions were like thousands of years ago. We can, age a log cabin for historical documentation. So dendrochronology has a really vast and really cool history within science. And so potentially so sagebrush does the same thing. Even though it's a shrub and not a tree, it grows rings every single year. So thicker rings mean that there might be more favorable conditions. So the plant is inputting more energy into creating more structure. And then more poor years, or when the plant's stressed, it might do smaller rings. So we can look at this difference in big rings versus small rings to answer a variety of questions. So what we wanted to see is, can we see climate stress in sagebrush by looking at the rings? And can we maybe discern when the sagebrush died? So calling back to earlier in this discussion where my advisor saw these big populations of dead sagebrush, can we try to maybe figure out when these sagebrush died and maybe why they died? Is it from water sequestration? Is it from drought stress? So these are kind of important questions. So now I'm going to be talking about the methods of how we're actually going to be looking at water stress within sagebrush using tree rings. And we're going to be counting and measuring rings by doing dendrochronology. So let me explain some dendrochronology using a really pretty sample. So we have this sanded and prepared specimen of sagebrush. And in that kind of yellow color, you can see the year 2017 of when the sagebrush was destructively sampled. And so 2017 was the last year that the sagebrush was alive. And then if you go through the rings, I marked decades and centuries and such like that. So 2017 is toward the top and then you go down to 2010, with a pencil, I did like a single dot. And then you go down to the year 2000 and that has 4 dots for a millennia, right? And then you go down, you can pretty much discern when the plant started growing. And it was around the year 1990, which is actually when I was born as well. So this is really cool. This is kind of an illustration of how we can count the rings in the beginning. So I learned how to do this at the North American Dendrochronological Field Week, or NADF. And this was held in Cody, Wyoming. I'm in the back right there with the plaid red shirt. And so this is a really important place where I got to learn how to do dendroecological techniques in a very short amount of time, in just 10 days. And I also met one of the people that helped out with this project, Amanda, as well. So this was a really cool thing to get me pushed into dendrochronology. So once I learned the techniques, I was able to go out with my advisor and some colleagues, and we went out to the University of California's White Mountain Research Center in Bishop, California. And in the late summer, this is when we collected our samples over a weekend period of time. So here's me with my advisor, Dr. Paul Shipman, with some sagebrush. So what we did was, over that weekend, we went to 5 different sites throughout Owens Valley. In the southern end of the Owens Valley, near Manzanar National Monument, I believe it's a monument, I'm not actually sure. So outside there was one of our field sites. And then as you go up, it goes more close to Bishop through the Owens Valley. And this is a massive area of all 5 of the sites. It looks kind of small from here, but on the left side, you have the Sierra Nevadas, which are huge mountains, like some of the tallest mountains in I think Mount Whitney being the tallest in mainland North America. And then mainland United States of America. And then on the eastern side, you have the White Mountains with the bristlecone forest, some of the oldest trees in the world. So from these 5 sites, we collected 11 dead individuals and 11 alive individuals from these sites. And so what we do is we'd find sagebrush that had pretty bulky stems, right, from living and dead individuals. And then we destructively sample them by using a saw, and then we quickly wrap them in cellophane so that we could preserve that chunk of wood and it wouldn't shift around. So sagebrush kind of did this weird, funky lobing thing, so sometimes they can kind of disintegrate on you. So this was important that we preserve their structure as soon as possible. And so then once we were we took these samples back to Cal State Northridge. We began the process of sawing them to make them more manageable. Then we would glue them onto plywood, these specimens, and then we would sand them with progressively finer sandpaper. We would start with a really gritty sandpaper and then work our way down to sandpaper that was so small it was on the nano scale. Very cool stuff. And here's Jacqueline, who did a huge amount of the sanding. Also, other people like Annie and Nick, undergrads at the time, as well as I was doing these. And this was in 2017, 2018, that we were processing these samples. The next step was me actually just reading the samples. And I got to teach other folks how to do this as well. Here's me looking at a scorpion under the dissecting scope, but normally I'd be looking at sagebrush samples under this dissecting scope. And by looking at these specimens under the dissecting scope, I would then judge them just qualitatively, right? Did they have a small ring or did they have a big ring compared to the rings that were to the left and right of them? Humans are really good at detecting patterns. And this is why this is usually one of the first steps in dendroecological work, is because humans are so good at detecting patterns of when there might be drought stress or some other environmental stress, climatic stress or something. We're really good at it. So you make these skeleton plots based on the lines, the solid lines would be for really small rings, but then you might put a B upside down for a big ring, right? Or then you mark where the sagebrush broke and you're not quite sure what's going on. But that break could be important as well. So it's important you record those as well. And the skeleton plot that I'm showing is from a dead sample. So I can't say what year it started and what year it ended, but I can give it's kind of floating in time, right? We're just kind of guessing. But we can compare different skeleton plots and kind of judge based on where the smallest rings all kind of have in common. So this is a bunch of different skeleton plots based on a master chronology I built using all these living samples, and I averaged them out and made a skeleton plot. And then just kind of guessing, using some like really easy averaging statistics, try to figure out like where I could put the other skeleton plots. So this was one scenario that I kind of built out of most of these samples are dead, so we don't know when they died and when they started growing. But the very top would be the master chronology, when I do know, because they were living samples, when they were born and when they died. So this next step it took a few years to get us to there, because life cuts in the way in graduate school. But this is where I actually measured the ring widths on a computer program called Coup Recorder. It's a really sweet program, not that expensive. And what it does is allows you to measure each width of the ring. And so what we did is we just did a single ray through each one of these samples. You could do multiple rays and then average them. We decided to just go a kind of quicker method. And this is something we can investigate in the future, like doing additional rays and seeing how that affects the results. I found papers that say usually one ray showed similar results in sagebrush. So we felt that it was okay to go through with this. So what we did was we measured single ray throughout the entire sample. And then the next step after Coup Recorder is a program called Dendro, and they work in pairs. And so what Dendro allows you to do is assemble all those measurements you made for different chronologies or time series, right? So I could do my living samples and my dead samples, organize them in Dendro, and I could spit out mean averages and stuff like that. And so from there, I have these chronologies that I've organized in Dendro. I can put them through what's called a Shiny app. It's this kind of statistical software that's been developed within R, which is like a language and a software that's publicly available. And I use a Shiny app that was developed from R, from DPLR, which is a package within R, called XDate R by Dr. Bunn. And it was a super cool program where I could cross-date the samples, where I could put those dead samples through statistical analysis, try to figure out when were they born and when did they die? What was the most likely scenario? And then for drought, I was able to collect data from NOAA. And so what you're looking at here is the drought severity index, so in the state of California. And so I was able to get this data from NOAA. So let's talk about the results. What did I find, right? So this comes from the Shiny app, where I was able to kind of put my living samples into a chronology of when they were born, and then obviously the year that I cut them down, right? So here's a time series from 2017 all the way down to the 1950s that shows my 11 samples. It should be 11, but it shows my samples of when they were most likely born, and so it's all different periods of time. My oldest sample was older than 60 years, which I thought was really interesting. But not totally surprising, as this is a pretty dry place. And like I said, the southern extent of their population, so they don't have, there's a lot of reasons why maybe they don't live into 100 or 200 years. And then what I was able to do was, from this chronology, and this is kind of a crazy plot, so let me kind of take you through this slowly. I was able to figure out the small years, the small rings, and then the big rings. So what you're looking at is all of my living samples, right? Or most of them. I think they're also spread out past on the y-axis, like even farther beyond there. But the important one to keep your eye on is that darker green one. That darker green one is the mean value for all these samples. And so here you're able to see if it dips down, that's when it was a dry year, potentially. And if it goes up, that's when it was a wet year or bigger rings, right? So that's really what it is. So the dips are small rings and the peaks are big rings, okay? So I was able to discern and averaged all my rings out and to create this master chronology, or this kind of mean value with this green line. So here we have And we have some years marked out as well. So in the year 2000, we have kind of a dip that you can see with some drought. And then if you go further along, we have 1975, or we have some dips as well too. And the further left you go, the older you get, the less samples get that age. So what's going on becomes a little, you can't say as much because there's not as much evidence supporting it. There's not enough samples supporting it. Still could be interesting. My other question, right, is trying to figure out when these dead samples died, right? So what we do is statistically try to correlate those dead samples within the living chronology. And this is a kind of confusing figure. On the x-axis, you have years, right? So so it's starting in 2017, and then potentially going down into the 1920s with some of my samples, potentially, and I'll explain that in a bit. So the top half of the graph is mostly going to be my living samples. And what the different colors mean, if it's red, it didn't, there wasn't a significant value for correlation to each other, but if it's blue, then there is a significant value for the correlation. So a few of my living samples correlated really well to everything else that was going on. But a lot of my other living samples, there's what we call noise. There was a lot of like environmental factors that could have been going on in their growth that were different, that didn't relate from one sample to the next sample. So the blue is kind of important thing here. So for a few of my living samples, they correlated pretty well, and they fit those dead samples in pretty well too to a time area. So this is the most likely scenario for when those dead samples died. And so you can see the important thing for here is going to be these time periods. So three of the samples died in the early 1980s. A few of them died in the mid 1980s. One of them died in the early 1990s. The mid 1990s, the late 1990s, and two of them died in 2008 and 2009. So these dead periods are important, right? And why are they important, right? Why would things die in these particular time periods? So next, I want to show you the results from the climate data, and this is just an example of the results because it's a big data set, right? And there's a lot going on. But this is just from 1997. And I told you a few of our samples, let's see how many it was. One of our samples died during this period. And so what we could look from our climate data, and I'll talk more about in our discussion and stuff, is like during this period, what I would do is I would look and see how was California doing at that time, or like around that time, right? Because there could be a lag period for when the sagebrush died, even if it's like slightly after the drought or just leading into the drought, like in the middle of the drought, right? But in 1997, we had a pretty severe drought in Owens Valley and Los Angeles as well. Let's talk about our discussion, right? So I showed you this kind of crazy graph. I showed you both of these graphs before. So the NOAA data down here, and then this graph that I made in my statistical software. And you see, I cropped them to be around the same time period. So here on the top graph is from 2017 down onward, and I cut it off about 1980. And then here, this goes from 2018 to the 1980s. And the cool thing to notice about this is the dips are about the same. So those droughts are about the same. So here on the NOAA data, if it's orange, that means it was a drought. It was a dry year. If it's green and upward, it's going to be a more wet year. And these match pretty well. So if you see here, there's some dips there, and there's some dips there well. And then it shoots back up, and it shoots back up here. It shoots back up here, and there's a dip right there. There's a dip right there. So let's be a little more specific. So around 2008, 2009, I told you there was a drought. That's approximately this region right here, and that's where we see this drought in this period. So 2005, 2006, we had rain, and then it went into a very severe drought right after. And that's when we had 2 of our samples die in that time period. So, and this is just our living samples. So our living samples correlate, and we need to do more statistical tests on this, but our living samples correlate really well with what's going on with California climatically. So every time there seems to be a drought, my samples, the living samples, dip down, and they show that drought by having smaller tree rings. This isn't too surprising. You see in the literature that sagebrush is used in some studies. It's not used in a ton of studies, but it is used in studies to show drought. And it does have a correlation to climatic data. And here you can just visually see that. It has the similar peaks and it has the similar drops from the NOAA data set from this Palmer Drought Severity Index. So that's an important thing we should investigate in the future. Like I said, it appears to have high correlation to drought, and that's just visually. So again, this needs more statistical analysis. The death years, right? I showed you that graph where I centered them. Here are they listed. So I had 2 samples that died in 1981, 1982, I had 1 sample, and that was a period of moderate drought. In the next series that died, 1985, 2 samples, that was severe drought. 1991, severe drought. 1997, severe drought. 2008 and 2009, another severe drought. So it appears this is the most likely scenario because, again, these samples are fairly young, so it's hard to put them within a period of time, right? I also want to be transparent and mention that I've lumped my samples together across sites. It's a pretty far-ranging area, so these are pseudo-replicated, right? But I have lumped them together, but this is just preliminary research. It does appear that the most likely correlation values that I was getting that were, not all of them were statistically significant, but most of my samples were statistically significant for this correlation. They do appear to match droughts, right? This would make a lot of sense if you think about it. I talked about how these plants are uniquely adapted for their environment, living in the desert, but they can be affected by drought because of their physiology. So another next step was we can kind of conclude that the we have more surety that these living samples are showing a climatic signal. They are correlating fairly well, at least just based visually on this NOAA data set. And it appears the most likely scenarios of when these dead samples died was during droughts. So we then want to look at, well, where are they accessing water from? From the Sierra Nevadas, water, the water table is really defined by snow melt. So a lot of that melted snow in the Sierra Nevadas goes down into the Owens Valley and builds up, and that's what a lot of the sagebrush depend on. So what would be really important is looking at the water table. Here's a map from Inyo County Water District that shows just during 2020, in spring 2020, the levels for water. So this data is available. We could attempt to look at what the water tables in all of these different wells was like during that time period. And we can try to see if we can correlate that to when these samples were dying. It's definitely possible. Another thing that we might want to further look into is increasing our sample size, especially for dead individuals, because it appears that there is a bunch of different events of when these sagebrush dies. It wasn't like a single event, though that drought in the 1980s seemed to kill a lot of them, 3 of them at least. So it'd be, if we increased our sample size of dead individuals, perhaps we could see more of them most likely died in the 1980s. Did something happen during that time period with the water table that might be of particular interest? We need way more buy-in and collaboration with the indigenous nations in that area. It's really important that they be involved in this process. So many of the activists are really passionate about water, especially after issues like Standing Rock. We really need to be working with them and have their permission and have an ongoing dialogue with the indigenous people there. It's critical. And that should be just all research period, should be with collaboration, consent, and communication with the indigenous folk. And we also need to be looking at the current history with water sequestration by the Los Angeles Department of Water and Power. I think that's really important that we try to, but before we make too many statements about like, this is their fault, this is our fault, or whatever, we need to be looking at what the facts are, what the history is. Did certain events happen at certain times? Did closures happen? Did they do take less water during this drought, or more water in the water table? What was going on and when? Before we make too many rash statements, but I think that's a critical piece of this story as well. So let's talk about some concluding remarks. Sagebrush in this part of its range is likely tied to climate. This is supported by the literature. This is pretty well supported just from this preliminary research, small sample size, but it's looking likely that it's pretty tied to climate and drought as well. Again, this is preliminary research into water, sagebrush, and environmental degradation. It'd be nice to continue this in some regard and to continue looking at how vegetation communities in the Owens Valley are being impacted by environmental degradation and taking out water from the valley and bringing it to Los Angeles. Sagebrush is likely a canary in the coal mine. I borrowed this from a colleague, an archaeologist. He's a really great guy. I'm really appreciative for his advice. And this is obvious to a lot of people, especially the indigenous folk that live in the area, that if the sagebrush is in trouble, there's a bigger issue that's going on. And so while we're asking like how much is this occurring, like folks already know on the ground. They know that stuff is changing rapidly and it's bad. But it's also important that we incorporate like this kind of evidence into it as well. Right, so we not only take account their stories and what they have to say in that sort of evidence, but we also build things like dendrochronology into that as well and strengthen these arguments for this environmental degradation that's going on. Water and people. So Owens Valley is rapidly changing. If not just the vegetation, the populations, ecotourism, there's a lot going on in that valley, and a lot's changing. And a lot of it's not good, and some of it's amazing. Some activism that's going on in the valley is really breathtaking for everything that it's gone through and suffered. And really, Los Angeles is changing as well. It is growing, and it is a huge population, and we are taking water not only from the Owens Valley, but also the Colorado River and from Arizona and the indigenous nations in Arizona. We need to find new water, and we need to find new water fast, because we are having Los Angeles and Phoenix, we are having huge, in San Diego too, we're having huge impacts on other places, on other communities that we've already been impacting severely. And so we have the brains, we have the technology, we just need to do it. And we need to have people supporting it, especially institutions like Los Angeles Department of Water and Power. They need to be increasing the initiatives for making eco-friendly desalination, trying to sequester water from the air, water table from Los Angeles itself, taking better care of the LA River. This is a systematic problem that I'm not going to have the simple answer for, and it's going to need to come from a lot of people. We need to work fast because there's a lot of damage that's already been done. All right. I'd like to thank Dr. Polly Schiffman, my advisor, for getting me involved in this research. It's been a lot of fun. I'd also like to thank the CSUN Department, Biology Department Outstanding Student Grant and the committee for funding this research and for funding me going to NADF and learning dendrochronology. Also, Dr. Jim Hayes, who is seen in this presentation, for his advisement and help with data collection. NADF, the North American Dendrochronological Field Week, I'd like to thank them. I can send them an email and ask them questions, a lot of the collaborators, and they've just all been really amazing. So NADF is a great tool, and I hope it continues onward. I also like to thank the White Mountain Research Center in Bishop. They were so nice to us, and the food was amazing. And it's a beautiful place. It has one of the only populations of Owens Valley pupfish. Go visit, go do research out of it. It's such a cool place. And I'd also like to thank the many collaborators on this project, like Jacqueline Fay, Nick Poulos, Annie Anderson, Amanda Huffner, and Mars Walters. Mars probably surprised that they're in this presentation, but they're always advising me and giving me great bits of advice and confidence. So because we had technical difficulties, I prepared this as a webinar, so I had this question slide, but I'd like to pose this on you. This is actually the Cal State Northridge campus. This is the orange groves, right? So orange groves were historically built in the San Fernando Valley after Los Angeles gained access to the Owens River. We have streets like Owens Mouth, right? To mark that. So orange groves are a part of the San Fernando Valley history because of this water sequestration, because of taking water. And we now at Cal State Northridge have one of the last orange groves in the San Fernando Valley. There's a few others across the San Fernando Valley, but we have one of the last ones. And in the middle of it, we have this gigantic water pond with non-native turtles, non-native mosquito fish, ducks that people just abandon. But there's other like native wildlife that interacts with this space. But it's quite a statement, right? We're watering these water-intensive trees with a giant water pond. We're one of the last ones that are having this. All the water moved 200 miles south from Owens Valley. And it's important we remember that. All right, thank you so much for coming. And I hope all of you have enjoyed this video.